Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology and will be taking the zoology part, that is animal science. My dear students, in the previous lecture, we came to know about different components of the immune system. We came to know about different cells which are present in the immune system which produce immunogenic response. We came to know that there are two types of immunities. One is which is present since birth, that is the innate immunity, which includes all the types of physical, psychological, okay, and cytological barriers, okay. We have started the acquired immunity in the last lecture. In the acquired immunity, we discuss about that it is the specific immunity or the adaptive immunity, while the innate immunity is non-specific, right? The characteristics of acquired immunity already been discussed. Now we are going to discuss about the complement system. What is this complement system? Complement system, it is the part of innate immune system, which can be activated by the adaptive immune system. Okay, so complement system is basically the part of innate immune system, but it is activated by the adaptive or the acquired immunity. It consists of complement proteins. There are various types of proteins which are present in this complement system. These proteins are synthesized by liver. These proteins are synthesized by liver. They serve as opsonins for phagocytosis. Opsonins means they help as a marker to identify non-self bodies. So these complement proteins, they actually help to represent the different cells of immune system, that okay, these cells are to be phagocytosed, they are not the part of our body. Okay, the literal meaning of opsonin is to make tasty reading for eating. Okay, so different types of phagocytes like natural killer cells, like neutrophils, like monocytes, they want to phagocytose it, they want to kill it. So opsonins, they actually help them to kill. Okay, they basically help them that this is the self part and this is the non-self part. Understood? So this is what the part of adaptive immune system. This phenomenon is of adaptive immune system or the acquired immunity, but it is helping the innate immune system to activate the complement system. Understood? They also lead to the formation of membrane attack complex. Okay, whenever any mucosal membrane is attacked by any antigen, the complement system get activated. Okay, and it basically gives signal to the phagocytic cells like natural killer cells, neutrophils, monocytes, macrophages to come and engulf them. Understood? The complement system, which is helping the innate immunity to go for, you know, phagocytosis, or to differentiate between the self and non-self. These are complement proteins. These complement proteins, okay, like you can see C5b, C7, C6, C8, C9. Okay, all these proteins, they are a kind of perforins. They create a pore, and because of this pore, the extracellular fluid, it rushes in, and the cells or the intercellular space will increase, which will further burst, okay, after reaching to the maximum pressure. So these complement proteins, they actually guard the plasma membrane, okay, to open the channels whenever the cell needs to be self destroyed. Understood? Now coming to acquired immunity. Acquired immunity is also known as the adaptive immunity. adaptive immunity or specific immunity. Understood? This immunity is specific and mediated by antibodies or lymphocytes or both. So this type of immunity is found only in vertebrates. Its response varies on the basis of pathogen or the challenge, stress. Thus, immunity recognizes the specific pathogen and works on eliminating it. Okay? So it basically acts because of production of antibodies or lymphocytes or both. Understood? It not only receives the victim of the infectious disease but also prevents its further attack in future. Means it helps to preserve memory cells. Okay, these memory cells help to remember the encounter. Help to remember the encounter. Understood? The memory cells, they are formed by B cells and T cells, and they are the basis of acquired immunity. These memory cells, they are formed by B cells and T cells, and they are the basis of acquired immunity. Okay, now coming to next, special features of adaptive immune system or the acquired immunity. First is specificity. This response is specific for different pathogens. Okay, so adaptive immune system or the acquired immune system, it is specific for different pathogens. It is not generalized. Okay, diversity. This system can recognize a very large group of or varieties of microorganisms. So this is diversity. So it is specific, it is diverse, it helps to discriminate between self and non-self. It can distinguish self tissues from non-self cells. Okay, mean the cells of the host and the foreign bodies like bacteria, virus, okay, the pathogens, they can be differentiated by adaptive immune system or the acquired immune system. In normal cases, no immune response is mounted against the self cells except autoimmune disorders. In normal conditions, all the organs of your body, all the cells of your body, they don't produce antibodies against themselves. Okay, but in case of autoimmune diseases, the condition can be changed. Okay, understood? Now, memory cells. In case of first time a specific pathogen enters the body, the adaptive immune system takes a longer time to recognize and to respond it because the pathogen is entering inside the body for the very first time, and that is why the pathogen needs to remember it. Okay, so this type of immune response is known as what? Primary immune response. And primary immune response is longer in comparison to the secondary immune response. So some memory cells are formed during such pathogenic attack. When the pathogen enters, there are some memory cells which are formed, okay, by the differentiation of B cells. And when a second time the same pathogen enters the body, the adaptive immune system responds very quickly because the memory cells, they direct, you know, they direct initiate the production of, or they direct signal the production of formation of antibodies, or they will signal the killer cells or the antigen presenting cells to work on. So these memory cells, they are basically, you know, are differentiated form of cell, and they are also known as primed cells. They are also known as. Okay. So when a second time the same pathogen enters the body, the adaptive immune system responds very quickly. This type of response is called secondary immune response or anamnestic response. Anamnestic response. Understood? So B cells, they actually differentiate to form memory cells or known as primed cells, having a longer lifespan. Understood? Now, these are the cells of immune system. Already I have explained it to you. Stem cell, it further differentiate to form myeloid progenitor means, and lymphoid stem cells. Myeloid progenitor simply means what? That these cells are precursors of myeloid cells. Okay, so myeloid stem cells and lymphoid stem cells. Myeloid stem cells, it can interconvert into lymphoid stem cells. These lymphoid stem cells, they further form premature B cells and premature T cells. Understood? So when we compare about the lymphoid stem cells, these stem cells, they form premature B cells and premature T cells. And a third category is natural killer cell. The natural killer cells is the third category. The premature B cell, they actually differentiated in bone marrow and the gut submucosa in mammals. So they mature in bone marrow and gut submucosa in mammals. Understood? The premature T cell or the T cell progenitor, it gets matured in thymus. It is of two types. Basically, it is of four types: T helper cell and cytotoxic T cell. Okay, T helper cell and cytotoxic T cell. There is two more categories: memory T cells and the fourth category is T suppressor cells. Understood? Now the B cell, premature B cell forms activated or B cell, which further form memory cell and plasma cell. Memory cells, they are known as what? Primed cells. Okay? Is it understood? The myeloid progenitor, it forms granulocytes, which further differentiate into neutrophils, eosinophils, basophils, mast cells, and the normoblast, it further forms what? RBCs. Okay? Understood? Platelets. So platelets, RBCs, dendritic cells, macrophages. The monocytes further differentiate to form dendritic cells and macrophages. Okay. Mast cells, basophils, eosinophil, neutrophil, they all are formed by the myeloid progenitor or the myeloid precursors. Understood? Now coming to next. Cell-mediated immunity, it is based on T cells. Four types of cells: T helper cells, killer or cytotoxic T cells, T suppressor cells, and memory T cells. Understood? So the acquired immunity is basically having two components. One is the cell-mediated immune system, and the second one is antibody-mediated immune system. Understood? This is the lymph and lymphoid organs along with immune system. The lymphoid organs and the immune system, okay, they produce different types of cellular components to fight against infections. We will be coming in the next lectures about the lymph nodes and the role of lymphatic system to provide immunity. Okay. Now the cells of adaptive immune system. T lymphocytes and B lymphocytes, they are the major cells of adaptive immune system. They are the subsets of or the subtypes of leukocytes. Okay. They perform different roles in the adaptive immune response. Based on these two types of lymphocytes involved in the responses, cell-mediated immunity and antibody-mediated immunity or humoral immunity. It is because of T lymphocytes. T lymphocytes cannot produce antibodies. They help B lymphocytes to produce antibodies. So the cells of adaptive immune system, T lymphocytes and B lymphocytes, okay. T lymphocytes forms the cell-mediated immunity and B lymphocytes, they form the antibody-mediated immunity or the humoral immunity. Now, coming next. In the initial stages, macrophage interact with pathogens and activates T helper cells by releasing cytokines, cell mediators, or interleukins or monokines. So the very first step is the macrophages, they interact with the pathogen. They activate T helper cells by releasing cytological mediators, cytokines, or interleukins or monokines. The activated T helper cells stimulate the killer T cells or TC, T helper cells TH, and the B cells which start dividing. So when B cells they start dividing, they differentiate to form the effector B cells, which are known as what? Plasma cells, and the primed cells known as the memory cells. So the very first step is the natural killer cell. It interacts with pathogens and activate T helper cells by releasing cytokines, interleukins, and monokines. The T helper cells stimulate the T killer cells and the B lymphocytes or the B cells will start dividing. Helper T cells produce lymphokines. T helper cells produce lymphokines, a type of messenger molecules. Immune system cells can interact or detect the lymphokines and follow the path to locate of their reproduction, which is the site where their presence is required. So T helper cells, they produce lymphokines. Okay. Immune system cells, they identify the lymphokines and follow the path to the location for their production. And these immune system cells, they detect the lymphokines and they follow the lymphokine secreting path okay, and they reach to the location. The lymphokines also aid, means also help B cells to produce antibodies. The lymphokines, that is a messenger which gives B cell a green signal to produce antibodies. Killer T cells or killer cytotoxic T cells destroy the virus and the cancer infected cells or the tumor cells. So killer T cells or cytotoxic T cells, they are basically having antigen receptors like this. These antigen receptors, they can attach to the surface of virus, okay, virus, okay. So these cytotoxic T cells or killer cells, they have antigen receptors on their surface which helps them to connect with the virus cell. The moment it happens, it will create or it will secrete perforins. Because of perforins, the channels or the holes forming proteins, perforins, they help to open the plasma membrane channels, and that is why huge amount of fluid from extracellular enters the intracellular, thereby increasing the size of cell and it will burst out. Okay. So the T killer cells or the cytotoxic cells, they have antigen receptors on their surface which helps them to bind with the membrane of pathogen like virus or a tumor cell, and they will further create the perforins, which perforins open the pores and the extracellular fluid, it enters inside the intercellular fluid and increasing the size of the volume, thereby bursting it. T suppressor cells suppresses the functions of cytotoxic T cells, means T suppressor cells help to maintain the activity of cytotoxic T cells and T helper cells. B cells also affected by suppressor T cells. Okay. Memory T cells are not involved in the killing of the pathogen but are involved in retaining the memory and aiding the secondary immune response. Understood? So these are the functions, different functions of cytotoxic T cells, T helper cells, T suppressor cells, okay, and memory T cells. Suppressor T cells, also known as regulatory T cells or TR, okay, they help to suppress the function of cytotoxic T cells and T helper cells and also maintains the activity of B cells. Understood? These are four kinds of cells. T helper cell, which basically initiate mean the macrophage. It interacts with the pathogen and activates T helper cells. T helper cells secretes lymphokines or the cell mediators. These lymphokines, they act as a messenger and they directly been attacked or identified by the immune system cells, and they follow the path from where this lymphokine is coming. The lymphokine also help B cells to produce antibodies. Cytotoxic T cells, they destroy the virus in tumor infected cells because as they have antigen receptors on their surface, these antigen receptors, they help to connect with the virus or the infected cells and bursting it by creating the pores. Understood? Memory T cells help in the secondary immune response. Now, for natural killer cells, I have already told you that natural killer cells, they attack and destroy target cells as they have phagocytic action, okay? And they are basically a part of antibody dependent cell mediated cytotoxicity. So natural killer cells, they actually are involved in the antibody dependent cytotoxicity. They can also attack parasites which are much larger than bacteria. The natural killer cells, they help to kill parasite. They help to maintain the cell-mediated toxicity, cytotoxicity. Okay. Now coming to next, antigen-presenting cells. Antigen-presenting cells Antigen molecules are processed by the antigen presenting cells like macrophages and B lymphocytes. So macrophages and B lymphocytes, they are antigen presenting cells. Okay, these antigen presenting cells, they help to present antigen to the antibodies. Understood? Antigen presenting cells, they are commonly known as APCs. It includes macrophages and B lymphocytes. Okay, macrophages are usually found in a resting state. Okay, their phagocytic capability is increased when they are stimulated. Okay, the macrophages, they are in resting state, but they get stimulated to become an activated macrophage. The macrophage are present along with lymphocytes, okay, in all the lymphoid organs. Monocyte is a blood macrophage and histocyte is a tissue macrophage. So macrophages, monocyte is the blood macrophage and histocyte is the tissue macrophage. Understood? These antigen presenting cells they help the processed antigens are presented on the surface of these cells. A T helper cell may recognize the antigen and become activated. So T helper cell, on recognizing the antigen present on the antigen presenting cell, and becomes activated. Antigen presenting cells, they can engulf antigen and present a particular fragment or a part to the T helper cells. Okay, so that is why they are known as what? Antigen presenting cells, and they are helping to present antigen to the T helper cells. Okay, T helper cells activate the B cells and killer T cells. So T helper cell activate B cell and killer T cell. These cells turn develop clones by frequent division in themselves. Antigen presenting cells, they can, divide and they form large number of cells. This is a response which is developed during the formation of cell-mediated immunity. Okay. The control of intracellular pathogens. Exposure to intracellular antigens expressed on the surface of a cell infected by a virus, bacterium or parasite, also called as antigen presenting cells. First step, a T helper cell binds to major histocompatibility complex antigen. MHC antigen means it is a complex of major histocompatibility complex, means the immune system and the antigen. Okay, on the surface of infected cell, activating the T helper cells with the help of release of cytokine receptors. So the very first step is APC. They present the antigen or a fragment of antigen to T helper cells. T helper cells, they are basically attached to the major histocompatibility complex, okay, and they get activated to produce cytokine receptors. These cytokine receptors, especially the lymphokines, are basically identified by different cells, immune cells like macrophages, which further increase the phagocytic activity. Then CD8 is cluster of differentiation, mean cluster or the group of differentiated cells, immune cells. So these differentiated T cells become cytotoxic, okay, and able to induce cell-mediated immunity. Apoptosis is the natural cell death. Understood? So the T cells, they release cytokines like lymphokines, monokines, and interleukins to basically activate the macrophages, and macrophages further activate, or it can also directly produce cytotoxic or stimulate the cytotoxic T cells, okay, to form a trap to kill the foreign bodies. The T cells, they also help to activate the antibody mediated immune system, that is the B cells. Understood? Cytokines transform B cells into antibody for providing plasma cells. So because of cytokines only, the B cells differentiate to produce the plasma cells and the memory cells. Okay. Some T and B cells differentiated into memory cells that respond rapidly to any secondary encounter to produce an amnestic response or secondary response. Okay. The humoral immunity or the antibody immune system, the extracellular pathogens or the antigens, in which it controls the freely circulating pathogens in blood, and the cell mediated immunity, it controls the intracellular pathogens. So cell mediated immunity, it works inside the cell to kill or to control the intracellular pathogens, and humoral immunity or the antibody immunity, it controls the pathogens which are freely moving in blood. B cell binds to the antigen for which it is specific. Okay. T dependent B cell requires cooperation with T helper cell. B cell proliferates and some progeny become plasma cells and memory cells. The plasma B cells or the effector cells, often with stimulation with helper T cells, produce plasma cell which further produce antibodies. Okay. Plasma cells proliferate to produce antibodies against an antigen. So this is the differentiation between the cell-mediated immune system and the humoral immunity. Okay. This is the difference between cell-mediated immunity and humoral immunity. Now there are other category of antigen presenting cells. Example, dendritic cells. These cells are characterized by a long cytoplasmic process. They look like this. These are the cytoplasmic processes. Okay. These are the different cytoplasmic processes. Okay. Their primary role is to function to maintain the effective antigen trapping and antigen presenting cells. So dendritic cells, their main function is to increase the efficiency of antigen trapping or antigen presenting cells. This is the nucleus. Okay. Okay. So they help to basically increase the efficiency of APC, antigen presenting cells. These cells are non-phagocytic. They are non-phagocytic. Understood? They are found in lymph nodes, spleen, thymus, and skin. Found in lymph nodes, thymus, spleen, and skin. There are different types of dendritic cells. There are further different types of dendritic cells. So these dendritic cells are the type of antigen presenting cells. So antigen presenting cells along with macrophages and the B cells, dendritic cells are also there. These cells have long cytoplasmic or protoplasmic processes. They will increase the efficiency of antigen presenting cells if they are non-phagocytic. They are found in lymph nodes, spleen, thymus, and skin. Okay, I'll write here. The type of dendritic cells. First is Langerhans cell. Langerhans dendritic cells, they are mainly found in the epidermis of the skin, which helps to trap the organisms coming in contact with the body surface. Present on skin and helps to trap the organisms coming in contact with body surface. Understood? The dendritic cells in spleen. The second category is dendritic cells in spleen. Okay. Then follicular dendritic cells in lymph nodes. Understood? Coming to next. Steinman was awarded Nobel Prize in 2011 for the discovery of dendritic cell. Okay. Was awarded Nobel Prize. For the discovery of dendritic cell. Understood? Now, there is a difference between humoral and cell-mediated immunity. Humoral immunity, main cells involved, B lymphocytes, T lymphocytes. Where do cells develop? Produced and mature in bone marrow. In bone marrow, both the types of immune cells are matured, both humoral and T cell. Produced in bone marrow, but in case of cell-mediated immunity, the final maturation takes place in thymus gland. Humoral immunity, it involves production of antibodies. It does not involve the production of antibodies. The pathogens are identified in case of humoral immunity by antigens floating in the blood, by antigens on the surface of infected cells. Okay. So cell-mediated immunity, it basically identifies the pathogens with the help of antigens which are present on the infected cells. Okay. And humoral immunity, it helps to identify the pathogens when they are floating in the blood. How are the pathogens killed? By the production of antibodies, by the specialized killer T cells or cytotoxic T cells. How do cells divide once they are stimulated? Cells divide into either plasma cells or memory cells. Cells divide into different types of specialist T cells. Okay? Now coming to next, the antibody immunity or the humoral immunity. This is known as what? Humoral immunity. Okay? Now, it is based on B lymphocytes which secrete antibodies. So B lymphocytes, they secrete antibodies. They are basically expressed by the surface presence of immunoglobulins or immunoglobulin molecules, okay, which mainly function as B cell antigen receptors. The immunoglobulins, they help to produce immunoglobulins, okay. Antibody, an antibody AB is a large Y-shaped protein produced by B cells, also known as immunoglobulins. Okay, it is used by immune system to identify and neutralize the pathogen such as bacteria and virus. Understood? So the B cells or the B lymphocytes, They secrete antibodies. An antibody is a large Y-shaped protein produced by B cells. It is used by immune system to identify and neutralize the pathogen such as bacteria and virus. Antibodies, they are also known as immunoglobulins. Also known as immunoglobulins. Antibodies have two heavy and two light chains based on the number of amino acids present in the chain. Antibodies, they have two heavy and two light chains based on the number of amino acids present on the chain. These antibodies are also known as immunoglobulins. But one thing remember please, that all antibodies are immunoglobulins, but all immunoglobulins are not antibodies. Immunoglobulins. Understood? So in antibody-mediated immunity or humoral immunity, it is very important to remember that it is based on B lymphocytes which secrete antibodies or immunoglobulins. This immunoglobulin is a Y-shaped protein, okay, which helps to neutralize and to kill the bacteria and virus. Neutralize, not kill, neutralize and identify. Antibodies, they have two heavy and two light chains based on the number of amino acids. Each chain has have some constant region. Mean the amino acid sequence of this region is same for a particular type of antibody. For a particular type of antibody, the constant region which is present on the heavy chain and the light chain both, it basically reflects that the amino acid sequence of this region will remain same. Understood? Each chain has a variable region at one end. Each chain has a variable region at one end. This region is responsible for antigenic recognition diversity. So, which part of the antibody is helpful to recognize the modulating or changing antigens? The variable region. Okay? So the constant regions in which the amino acid sequence will remain same, but the variable region in which the amino acid sequence will change, and they will identify different types of antigens accordingly. Understood? Now next, each tip of the upper tips of the Y of an antibody contains a paratope. Paratope means what? Analogous to lock. The paratope is a type of lock that is specific for a particular epitope, mean analogous to key. So as you know that a lock is very specific to a key. So paratope is also specific to a epitope. Suppose epitope is an antigen and paratope is an antigen binding site. So antigen binding site will remain specific for the antigen. Okay? Allowing these two structures to bind together with precision. This binding mechanism is used to tag a microbe or an infected cell for the attack by other parts of the immune system. So basically antibodies or the immunoglobulins, they help to identify the antigen by making a lock. So these antigens, they have, the antigenic sites which are recognized by antigen binding sites present on the tip or the terminal portions of the Y of an antibody. Okay. The antigenic determinants of the antigen, they basically make a lock and key type of mechanism to fit in the antigen binding site of both light chains and heavy chains. I'll explain with the help of diagram. This is the antigen, okay, and this is the antigen with antigen binding site. So you remember, suppose This is the heavy chain and this is the light chain. I'll change the ink so that, okay. So this is the light chain, light chain, okay. And this is the heavy chain. So they have basically antigen, this antigen, this is antigen, okay. These are antigenic determinants, the processes, antigenic determinant. Okay, and these are antigen binding sites. Okay, so the antigenic determinant and the antigen binding sites, they help to bind the antigen to the light chain and the heavy chain. Understood? So these are the antigen binding sites. You can see the groups are here. So for every antigen, the groups are specific. So the heavy chain and the light chains and antibody molecule is made up of 4 peptide chains. There are 4 peptide chains. Two small called light chains. You can see the pink one. This is the light chain. This is the light chain. Okay. And the blue one is the heavy chain, two longer. So these are the light chains which are shorter. This is the light chain. This is the light chain. While the continuous, they are what? These. They are heavy chains. They are heavy chains. Understood? Now, it is represented as H2L2. Two heavy chains and two light chains. The heavy chains have larger number of amino acids while light chain has smaller number of amino acids. Heavy chain and light chains may be either lambda or kappa type. Type of heavy and light chains, either lambda or kappa. Okay, lambda type or kappa type. Understood? Now you can see the variable region. This is the variable region, okay, which have the capacity to change the sequence of amino acid according to the antigenic determinant or the antigens. Okay, so these are the variable regions of heavy chain. Okay, variable regions of the heavy chain. These are the variable regions of the light chain. Constant region of the light chain, constant region of the heavy chain. Same case for here. Okay, they are having a disulfide linkage. You can see the disulfide bonds, disulfide linkage. Okay, and this portion, the tail of the Y, this part, the tail of the Y, these are the ends of the Y. Okay, this is the tail of the Y. So it is the effector region. This is the effector region. Understood? You can see that there is a hinge which is present, which basically connects both the heavy chains of both the sides. Okay, now the disulfide linkage help to connect two light chains with heavy chains. This disulfide linkage is basically helping to connect light chain to heavy chain, light chain to heavy chain. Understood? Two sulfide bonds also link to heavy chains. These two sulfide bonds, they are basically linking to two heavy chains. So light chain, the disulfide linkage, the disulfide linkage, it is pursuing two functions. Helps to connect light chains to heavy chains and two heavy chains. Understood? The part of the antibody which is basically maximum flexible, it is called the hinge region. It is known as the hinge region. These projections, they are known as what? Arm. This is the hinge part, this is the hinge part, and this is the tail part. Okay? Now, there is a very important region which is known as what? Fragment antigen binding, FAB, okay, and fragment crystallizable, FC. It is not mentioned over here, okay. I'll mention fragment antigen binding. You can see the antigen binding site, this FAB, fragment antigen binding, and fragment crystallizable, okay. These are the two identical fragments of a Y-shaped molecule which having antigen binding sites and thus named the fragment antigen, okay, binding, FAB. The antigen binding sites bind to the specific antigen in the lock and key pattern. You can also see, okay, the lock and key pattern. This is the lock and key pattern. Okay, the specific grooves are there. Okay, which forms the antigen antibody complex. Understood? Now, there is a third fragment which lacks the ability to bind to the antigen and can be crystallized. There is a third fragment which is not having the capability to bind to the antigen, and that is why it is isolable, crystallizable, and it is known as what? Fragment crystallizable. So fragment crystallizable. Which is not able to bind. Not able bind to antigen. Understood? That's why it can be easily separated, crystallizing. So this is the structure of antibody, a typical antibody. Now, alternatively, this can also neutralize its target directly by blocking a part of a microbe that is essential for its invasion or survival. So antibodies, they can directly neutralize the target by blocking a part of the microbe which is essential for its invasion or for survival. The antibody has an Fc receptor, that is F crystallizable, at the ends opposite to the variable regions, at the ends which are opposite to the variable regions. Remember that, located at the base of Y, base of Y. That is why I have written here. Yeah, crystallizable, base of Y. Okay. It contains a glycosylated site. This region is involved in interaction of the antibody with other components of the immune system. So F crystallizable, it contains a glycosylated site which helps to interact the antibody with other components of the immune system. What are the actions of antibody? First is agglutination. The combination of antigen and antibody, it is known as what? Agglutination. Antibodies, they attach with antigen, okay, which is present on the surface of pathogen. So agglutination is the process of binding of antibody along with antigen. Opsonization, it is the attachment of bacteria, okay, with opsonins like IgG and IgM, promotes phagocytosis. Opsonization simply means it helps to read the bacteria or the foreign body. So attachment of bacteria means the antigen, Ag is antigen. with opsonins like IgG and IgM promotes phagocytosis. Is that understood? Okay. Now, neutralization. Antibodies neutralize the toxin of bacteria by attaching with them. The main three actions of the antibody immunoglobulins. Okay. First is what? The agglutination, means it is the binding of antigen with antibody. Then the second is what? Opsonization, attachment of bacteria or the antigen with the opsonins like IgG and IgM, which helps to identify. And the third is neutralization. Is that understood? So in the next turn, I will be starting with the types of antibodies. Till then, please go through the lecture again and again so that you can remember it. There are so many terms. Till then, bye bye, take care, stay safe.